morning, everyone. Thank you for that wonderful introduction. I promise only half of it's true, I think. Some numbers were doubled, some had it as zero. I'm a big fan. Well, first, thank you to BioForward for inviting me. Thank you to Illumina for sponsoring, and really it's a pleasure to be here. I'm going to go through a fairly large amount of topics in about 55 minutes. I'm a big fan of fairly preposterous talk titles. I think this one kind of fits the bill. So with this talk title, I can talk to you about pretty much anything, and I will plan to do that this morning. So first, I'll start with a bit of an intro and then go through his talk in 10 parts. And since I'm a programmer as well as a geneticist, of course, number one, it starts as number 0. So first is this idea that NASA has to put boots on Mars by the year 2035. We're fortunate enough to contribute with several grants and projects with NASA to contribute towards this goal. You can see here we want to get technology and science to the point where we can actually put people on Mars and have them safely land and return back to Earth. But eventually we want them to stay there. Now, in this rendition, it is unfortunate. It appears as if Mars is sucking all of the nutrients out of Earth to go in the, that's not the plan. I want to reassure you right away that the plan is to be on both planets in a sustainable fashion, to really be an interplanetary species. And so, but the way we have to study this and think about this, I'm going to come back to this at the end, because we have to really go back to the very first principles to think about what happens to the body in space, how can we measure biology in general, and how can we use all this information to begin studying long-term spaceflight. So I'll start at the beginning. The reason I became a geneticist, and why I think genetics is the best, is because the molecular recipe for the synthesis of all the cells in your body is present in that very first cell. So all of you in this room should have started out as an embryo with one cell. If any of you did not start out as one cell, please come see me after the lecture. I would like to do some studies on you, but you should have all started out as one cell, and all the instructions were there for the synthesis of your body. This is true whether you're a cnidarian or an invertebrate, like a Drosophila, or a vertebrate studying other invertebrates. Or in the case of some point in your life, you might necessitate huge amounts of ice cream. This has happened, for example, to my daughter, who here, I think somewhere in her genetic code is embedded this need for huge ice cream. And so the way that we normally study this, as geneticists, as scientists, as biologists, is, of course, we think about things through the central dogma. So DNA, RNA, protein. In this audience, we're all probably very familiar with this structure of how we study the mediation of information in life cells. But what's extraordinary is what's changed in the last 5, 10, and 15 years is that this static picture from the 50s and 60s is much more dynamic today. So instead of just trying to look at one assay at a time, we can now, in many instances, which I'll tell you about today and this morning, study all of them in some cases simultaneously, and certainly at much higher resolution with far greater depth than we've ever seen before. And so this leads to looking at DNA and RNA and proteins, and sometimes in real time, and leading to what is the understatement of the century, complex behavior, which is how you process food, information, essentially your mediation of your cells through the environment and space and time. So what has enabled this primarily has been this slide. And so if you've been to any genomics meeting recently, everyone is using this slide because it's really exciting, the fact that something that used to cost us hundreds of millions of dollars today to sequence a whole human genome, thanks primarily to Illumina actually, is now in the range of $1,000. So this has got everyone pretty excited because this is faster than Moore's law. It's really the fastest pace of any technological change that's ever happened in human history. So a lot of the excitement about the $1,000 genome, or just last year, this idea of soon being a $100 genome coming soon. It was very smart of Illumina to just say soon, and then their stock price went up. And just announced products coming soon, and then it worked very well for the stock. But extraordinarily, it's coming soon, so sometime soon it'll get even cheaper. And what this means is that the amount of data that we'll be generating is no longer astronomical, it's bigger than astronomical. So in this paper, there was a debate of what's the biggest amount of data, genomical or astronomical data. And at this current pace, we'll be approaching zettabytes or even soon yottabytes, 1 trillion terabytes of information within the next 5 years, basically, so 5 to 10 years. So what that actually means is every day that any geneticist wakes up, there is more information on that day than any other day prior. So it actually means that every single day in genetics, it's the best day to be a geneticist. So it's empirical enthusiasm. I'm not making that up. It's just a fact of just existence. So if I seem generally kind of happy when I wake up, it's because it's the just simple fact that the capacity for discovery is bigger and better than ever before. The sort of amount of information and the metadata around it is better. It doesn't mean you'll always have every experiment work. You won't have the best day in lab every day as a scientist, but the capacity has never been greater. So what this lets us do with this amount of data, this genomical amount of data, is actually to begin to study all the omes, and I'll lay this out because this is important for how we study astronauts. So the first ome that we want to look at is the epigenome. And so the epigenome is the regulatory layer behind DNA, how you package and contain DNA and regulate it. So in my laboratory, one of the simple things we do is try to really understand this question of how do you go from a simple methyl group, just add one methyl group to, say, a cytosine, and how does that lead to a phenotype? And how does it actually transmit information and regulate gene expression? So you can draw this number of different ways. You're familiar with this, probably, if you remember your chemistry. It's actually a very old modification, first discovered in 1925, this idea of methyl cytosine, or MC. And some of the very first studies on methylated cytosine came from 2000, actually noting what happens even during embryogenesis. So what happens when egg and sperm come together? You can see your sperm on top and egg on the bottom. And this is the antibody for methyl cytosine in green. And you might remember this. The way I remember it is like there was basically candlelight, and there was Marvin Gaye was playing, and there was wine, and that's how I remember my embryogenesis. I was too small to remember it really, but that's what I want it to be. I don't know if it is, but that's how I feel. But anyway, this is a mouse embryo, though. This isn't me. But what happens right after egg and sperm come together, it's this beautiful paper that demonstrated the paternal genome on top gets rapidly demethylated first. So you pull away all of the methyl groups to reset all of the epigenetic marks across the whole genome to enable that one cell to become all the cell types in your body. And so you can see here, eventually the maternal lineage also gets reprogrammed when you go from 1 cell to 2 cells to 4 cells. Eventually all the epigenetic marks, except for imprinted genes, get reset so you can become all these cell types in your body, what is called Waddington's epigenetic landscape, is that we all start out as a marble of totipotent cells and wind our way down lineage specification to become a very particular cell type. And so this has been known for several, really decades at this point, but something really new happened about 5 years ago was Hannon had a paper and then Steve Horvath had a paper. He just took all available data that had DNA methylation and age and said, I wonder, can we predict the age of someone based on DNA methylation? And this is across all different tissues. You can see here there's almost a linear ticking clock, essentially an epigenetic clock in all of our tissues that tells you how old you are. And so this is a really interesting metric, and this was replicated by other groups here. And so that actually means what we could do is if you don't want to tell me how old you are, actually, for example, if you want to swab your phone, one of the questions outside is, well, how old you are? And you say, well, I don't want to tell you how old I am because, maybe my birthday's tomorrow and I don't feel like telling you how old I'm going to be. But it doesn't matter, actually. What we could do is we could wait for you all to leave this room and then sequence the DNA on the chair, and then just basically I could send you like a creepy email and tell you how old you are, basically. So, and you might think, can you really do that? Is it that good? So one thing we did in the past year was we said, well, let's see, this hasn't been in the literature. Let's see about, could we make sort of a mock crime scene and with, sort of take donors of known age, put fresh blood samples across a car, and then put them into a plate, and then test them on a whole genome bisulfite as a method for looking at all the epigenome versus a targeted panel. And, let's see if we can get it right. And, place them in an actual sort of mock crime scene. And actually, we had controls in blue, and then the samples from the crime scene, fake crime scene here. And you can see we can actually predict pretty well exactly how old you are, within a few years. It's not perfect, but we can get a pretty good match of how old you are. So this does have forensic implications, obviously, but also it has some other maybe implications that we haven't seen yet. So, for example, when I was a kid growing up in Racine, Wisconsin, I wanted to get like a fake ID. Every 17-year-old wants a fake ID. You want to go check out that bar and go, you're just a young kid. What do you want to do? You need an older cousin. My older brother wouldn't give me his ID. He's now an upstanding citizen and a politician, so that makes it good. But he's the mayor of Racine, actually. So, so he didn't give me his ID. He was actually an upstanding citizen. But today, it's much easier. Like you can go to YOLOIDs.com, and you can get a fake ID for any state in the union. And I have no financial relationship with them. I just think it's interesting that this problem is now solved for the next generation. Yes, you can pay by Bitcoin. You're probably wondering. Yes, you can. And so you could imagine in the future, you're going down State Street and you want to get into a bar, and then maybe they say, well, listen, I'm going to check your ID, but I'm going to look at your epigenetic age, and I don't know if you're old enough to get in this bar because your epigenome is too young. Now, could this happen? I don't know. That's the end of my social commentary for this morning. But basically, this idea, it's not impossible, but there's one thing you could say if you're in front of that bar. You could say, well, wait, bouncer dude, I can tell you I've had other reasons why my epigenome changed. There's epigenome dynamics that can and do occur, and in particular, in a more serious note, what we've published on is in leukemia in particular, you can have an acceleration of the epigenetic age. In some rare cases, it can even decrease. In some cases, environmental exposures can also change it. So we know that in leukemia and also in other cancers, you can have relapse occur. And one of the big questions we study as well is what drives relapse in cancer? Is it genetic? or is it epigenetic? What's driving it? And actually, what we published in this paper is that it is often both, or in some cases, if it's not one, it's the other. And so you can actually have really two different paths for evolution, at least in leukemia, and also we've seen in glioblastoma. And so what we developed to study some of these questions are open source tools, open source packages. One is called MethylKit, and one is EDMR. In lab, originally we called this like a laboratory toolkit for methylation analysis. We called it MethLab, but then we thought it'd be a poor choice for an algorithm, so we changed the name of it to MethylKit. So we didn't use that. But what we do have now is something called EpiSuite, which is MethylKit, EDMR, and MEthClone, a variety of packages and C++ tools to analyze epigenetics data. And so what we use these tools for, particularly in leukemia, as a quick vignette, is to think about, again, this question of clonal selection at the epigenetic level. We often think of resistance. You think of antibiotic resistance. We think of resistance to therapy genetically. But we wanted to see, is it also true, can you have epigenetic selection and resistance? And then take patients we couldn't discern whether they're high risk or low risk, and can we separate them out using these epigenetic markers. And here for the first time we showed patients that had no prognosis, you actually could separate them out. So this is on the epigenetic side as a quick vignette, and I'll come back to that later. Now the other thing that is, of course, part of the omics that we have to study is the transcriptome, which of course is the RNA here in the middle, and how genes get activated and expressed. And what's interesting is in the year 2000, the estimate was that there might be 120,000 genes in the human genome. This is before the genome was completed. So this was, we now know, was an overestimate, but it's interesting if you look at what's happened over time, or really how many genes are there today in the human genome. We know now there's about 20,000 coding genes, but there's actually more non-coding genes than coding, those that don't make protein, those that actually are functional in their own capacity and doing regulatory work, sometimes secondary structure. And there's also, of course, a lot of pseudogenes and other immunological related genes, but there's actually about 60,000 genes in the human genome. So, you should never be talking to a reporter or talking to someone, say, or a holiday, you're going to Thanksgiving pretty soon. Someone will say, well, I study this one gene out of 25,000 genes in the human genome. Don't say that. You're actually lying whenever you say that. There's actually 60,000 genes. And even that number is not static. In the past 7 years, we've discovered thousands of new genes. You can see back when I started my lab, we had 50,000 genes. Today we've got about 60,000 genes. So the fundamental process of discovery and annotation of the human genome, one of the best studied genomes that we have, is still ongoing. A lot of it is these non-coding RNAs you can see here in green. We're still finding them. We're still discovering them. So again, this part of discovery that's still happening is really an active part of genetics. It's not done yet. And this means actually by the time we get to Mars, we might have 100,000 genes. So that's kind of good. I think we'll need all the genes when we get there, or at least to know where they are. And so this process is still ongoing. So this, which is exciting. The other ohm we're going to keep an eye on is the epitranscriptome, which is behind the RNA, the regulatory layer behind RNA, which is fundamentally built on the same question. If I add a methyl group, can I add it and modify sort of one of the bases of the genetic code and actually modulate the function of an RNA by adding, in this case, methyl-6-adenosine and modify it. So this is also very new research, but just the RNA code is just now coming into focus. But one paper we just published last year is actually looking at when you modify RNA, what does it actually do? So you think of RNA as genes being expressed and they're on their way to get transcribed or being regulated. But here, if you actually add the methyl groups, you can see our little M6A plots here. In this case, for like MYC and BCL2, really powerful oncogenes. This will actually change whether or not you have translation that's efficient or not, and maintain the stem cells inside your bone marrow. Whereas if you have mutations in METL3 or you remove it, it dysregulates this epitranscriptomic mark and changes the translation efficiency and leads to apoptosis. And so it's interesting that another sort of lever of gene regulation is not just that whether something gets turned on or off, but how you decorate the RNA, just like you think of DNA. You wouldn't think of DNA without thinking of how is it packaged, how is it modified. The same is also true now of RNA. So this epitranscriptome is also part of the ohms that we study. And so to jump back to the central dogma, to close out the first part of the lecture, is we don't think of it just as a simple linear trajectory anymore. It's actually, there's many types of guide RNAs. Information can flow back on itself. DNA transposons can copy themselves. Reverse transcription can move information backwards, and also RNA binding proteins can move information backwards. Ribozymes can copy themselves, and prions. Some of you maybe don't eat beef anymore because maybe you're afraid of getting mad cow disease, because it's kind of creepy, like a protein touches another protein and copies the information. Kind of freaks me out a little bit. But there's other places in the central dogma that self-replicate. And so it's not just prions. There's a sort of equally distributed creepiness across the entire central dogma you can see here. And so there's also the epigenome, there's the epitranscriptome, all post-translational modifications you could call the epiproteome. And we know that some of these epigenetic marks are also inherited. They transmit through generations, especially it's been established in mice and potentially in humans. There's even viral-induced microRNAs that can transmit between generations in C. elegans. And also prions can even go between evolution species. And so this is what we say, when we think of any problem in the lab today, this is basically how we imagine it. When someone says, well, what's happening inside the human cells? It could be in any one of these levers. And so that's the human cells. And again, all of you have these human cells. This is what's happening inside human cells. All of you have about 10 trillion human cells. Some of you more, some of you less, but you all look great today. But you all have about 10 trillion human cells. But there's other reasons why phenotypes can and do occur. It's not just the human cells. There are, of course, the others. So we get all excited about sequencing human genomes. It's getting cheaper. It's wonderful. But there's more than one genome in your body. So in the cellular democracy of your body, your human cells are actually a minority party. So if there was an election to see where you should go, you would never win the election. You would just jump into a big hot tub full of yogurt, perhaps, to hang out with the bacteria. Maybe you do that anyway. That's your own prerogative. That's what you do at home. But if the vote was to see where do I hang out with, the bacteria would always win. And essentially it's because you're outnumbered anywhere from 1.3 to 4 times as many of bacteria to human cells and more active molecules in the gut than there are human molecules. What this looks like is big, large eukaryotic cells are human, and you're covered, all of us are covered with these bacteria in and on and around us. If you step onto a scale, it'd be several pounds of your body mass. It would actually be these bacteria. So if your diet is not working, you can legit blame the bacteria and say, well, my bacteria, I got a lot of bacteria today. You can say that, not only because of their weight, but also what their biochemistry is, which I'll tell you shortly. This has led to a lot of work called the Human Microbiome Project, just to map what are the normal flora, just in different parts of the body, vagina, stool, essentially tongue, nose, different areas of the body. Every part of our body is colonized by this little ecosystem that's just called the microbiome, which also is part of your phone, which you'll see later today. But this led to a lot of exciting work to map what was present in normal bodies and then link it to interesting diseases. So it's been linked already to obesity, how you process food. It's been linked to inflammatory bowel disease and also Crohn's disease. And so far, over a dozen different human diseases, we think of human disease, are linked to and likely mediated by the microbiome. So some make obvious sense. You think of eczema, you think of even cavities, but ulcers we used to think were caused by stress. But now it's Helicobacter pylori, and so we can understand the context of ulcers and gastric inflammation. But autism, depression, things that you wouldn't normally think of but are now linked to the gut-brain axis, and we know are probably mediating or in some cases exacerbating some of the phenotypes that we see in patients. So this led to an explosion of research at the NIMH for looking on the microbiome. And also in cancer, so we think of cancer like HPV or HCV as leading to cancer, but one of the more striking papers from last year is to think about, could you have bacteria hiding inside of a tumor? The answer is yes, at least from this data. And what you may have to do first is actually treat with antibiotics and then treat with chemotherapy, in this case, to actually treat the bacteria inside the tumor. So again, surprising places, you'll find them everywhere. And there's been other surprising things that have come out of the microbiome space. This is more on the disease side. But on the healthy side, what if you could just replace your skin microbiome? What if you don't like the microbes on your skin, you want to get some new ones? What if you just don't like showering anymore? Maybe some of you don't like to shower anymore. You can actually take this bacteria and spray it on your body because it's ammonia oxidizing bacteria. And so you don't even have to shower. You don't have to use soap anymore. The CEO of this company has notoriously not showered for over 10 years. And he doesn't smell at all, a little bit greasy, but otherwise doesn't smell. But he's covered, his skin microbiome with this AO biome, and so you can read a first-hand reporting of it here from a New York Times reporter. And so this is kind of exciting. It's almost like a superpower. You get like a superpower skin microbiome. What other things can the microbiome do? In some cases, they can work on your behalf, like you don't have to ask them to do so. This is work from Michael Fischbach and Mohammed Dani, and saying, okay, we've got all this Human Microbiome Project data. What if we could just go in and mine all the sequence data and look and see, are they making something new? Can we find a biosynthetic gene cluster, or it's a cluster of genes together in one contig that are making one sort of molecule, or BGC, and predict a new antibiotic they called lactocillin, and see can we validate it and see it's actually working. They did validate it. It showed actually was a new antibiotic that they just found from normal vaginal flora. Just in normal people, basically, you don't have to ask them to do it. The bacteria are already defending you, sort of giving you a protective vaginal microbiome force field, it's defending you against other invading pathogens. Now, that's not a technical medical term. I'm going to tell you the protective vaginal microbiome force field has not become on the market. But conceptually, it's not just a vaginal microbiome. It's a skin microbiome. It's the gut microbiome. Every area of your body that has a healthy commensal ecosystem itself is defensive against other potential pathogens. And so this has led to this idea of could we find new antibiotics in the microbiome? Could we engineer commensals to be more protective? The answer is to a large degree yes. Or what if you have some dysbiosis in your gut? Could you almost use a microbiome like a transplant? So in this case, this has led to an entire field called fecal microbiome transplants, which if you've not heard of these, they're exactly what they sound like. They could have called it gut transfer. They could have called it gut health. But what ended up in the literature was FMT, for the term. Basically, this is a way to treat if people have persistent diarrhea I won't go away from a C. diff infection. This is one way you can actually transplant the entire ecosystem from one person into another. It's an FMTs. And this has led to really interesting also treatments, but also interesting headlines. So this is a really great headline, which I think is unfortunately titled, Gripping Fecal Transplants. I think could have called it something else here. The other thing that's amazing is people have heard about this. So patients, I'm a big fan of patient advocacy and patient empowerment, and patients often will make their own websites and say, well, I've heard that Jane has got a really good FMT that she gave to 3 other people, and they all were successful. So can I get some of Jane's secret sauce? Can you send me a sample and just send it over here? So patients started to connect to each other, as people will do, and they launched a website, which I couldn't make this up if I tried. It was called thepowerofpoop.com. Which is just what it sounds like, power poop. And so in this case, you can go to the website and it's like, what's the rhythm of your gut? Tell me about what is C. diff? How do I treat? What about came out Tuesday? Can you have an improved? Are you a super pooper or a regular pooper? There's all these discussions on here. Or instead of repopulate your microbiome, they'll say repopulate. There's all these great jokes on this website, but they're not crazy, right? They're saying, okay, I'm going to send you a stool, but there's actually a 95% success rate for C. diff and a good success rate for ulcerative colitis. So almost nothing in medicine has a 95% success rate. So it's really an extraordinary treatment efficacy. And so this led people about 4 or 5 years ago to say, okay, we need to start banking samples, getting a big biobank. And so Eric Alm launched something called Open Biome. And so more and more people started banking samples, but they needed samples and they needed some money. And so they said, let's do a crowdfunding campaign. And when they had some great graphics that were like, listen, this might be the most important thing you do all day. Look at how many people you can treat, depending on your volume. What you just throw this away. What if you do later today at the conference? You should flush and think a little bit sad and be like, what if my medicine, what if I just threw it away? Because you don't know what you're doing today. This could be the most important thing you do all day. So they said, okay, we need a good graphics. They got that. And they said, we need a good name for our campaign. So they called it the CARE campaign, which I thought was great. So you could donate money, donate samples, and then they said if you're regular enough, you can even get paid. So you can make up to 13 grand a year. If you're a grad student, this is pretty big money, right? So this is good, right? So they started getting cat samples, dog samples. It got out of control. And so I was emailing with Eric about this, and he said, yeah, it got out of control. But now you have to live in Boston. You have to be regular every day. You can't just send them all your supplies. So it's much more regulated now. But they've also spun this out into a company. But they also, as a policy question, what's interesting is what I've told you is that basically the microbiome makes drugs and processes drugs, and so it's really more like a tissue and a drug. But how do you regulate something that does both? So far it's been regulated more as a tissue, so it's much easier to donate, which has been very good, I think, for patients and for the industry. So that's kind of the superpowers of the microbiome. You can do all these things, get force fields, you can treat people. But generally, if you say bacteria, or you say it in the subway, for example, people are just afraid of touching any surface. And so in this case, you can see I caught a woman here and riding the subway, and she's terrified to touch the steel railing. But it's interesting is the aperture and the holes of that paper towel are bigger than most bacteria. So most of the bacteria will just go right through anyway. I don't know if she knows that. I saw one woman do an upgrade and got a lab glove on. She's like, fine, I'll do this. Riding the subway, just terrified to touch the steel railing. So whenever possible, we shouldn't be afraid of things. We should replace fear with knowledge of what's actually there. And so, because if you're fearful, you might become other things. You might start to get superstitious. So maybe you only ride the subway on Sunday nights, or maybe you'll only ride with a certain lean with the butt method, so you don't touch anything while you're reading. I've seen people do that. Or maybe you start to get cruel and be mean to people who look like they're leaving bacteria around. And then, to conquer fear is the beginning of wisdom. Bertrand Russell's my favorite philosopher. So I think, we should start to conquer this and see what's there. So that was one part of my interest in microbiome work about 6 years ago. But then also, every time we'd sequence a human genome, we always had fragments of DNA that didn't map to the human genome. And so some of this is because the human genome is incomplete, but almost always it's because we have bacteria and viruses everywhere, which just kind of bothered me. I wanted to have a fragment of DNA. I wanted to know where it came from. I'm a geneticist. It's like my job, right? So I didn't know where it came from. That was happening. And then the second thing that happened is I just became a father, as I again shamelessly used my daughter. And when she got old enough to ride the subway, one day she was actually grabbing the pole, and she actually then licked the subway pole. I didn't get a picture of her licking, but this was her shortly after, and then she once dropped a sandwich and ate that off the floor. But the licking moment was one of those moments, if any of you are parents, it's just a moment of pure terror and fear of something has just happened. And I wanted to know what it was, the licking, and just a lot of things had just occurred. So I'm a dorky geneticist. I go home, I look on PubMed, Google Scholar, and there must have been a study about what's on mass transit systems. There hadn't been a single study. There's 2 studies about what's in the air by Norman Pace and Patrick Lee, but nothing about surfaces used in New York City by 5.5 million people every day. We have no idea what's there. And I thought, well, that's strange. And so in a world of sort of lack of information, we just began to collect samples all across New York City and said, what if we could build a city-scale genetic map of the metropolis, or what the New Yorker called a metropolome? Could we build a metropolome? So we set out to do this about 5 years ago and published a first study 3 years ago, which is to sort of get a city-scale metagenome profile. So we sent out students across, and basically the background of the students, there were 5 students that came in there. I said, they were looking at genetics and cancer and different diseases. I said, well, those diseases aren't going anywhere. How do you feel about swabbing the subway for the whole summer? And they were all really excited about it. So it was great. It's a perfect summer student job. And we went out and swabbed. We built up an app. So actually, as you enter the data, actually it's what they're using also outside. It's just a geotag and a timestamp, populated on the map, and shotgun sequenced it on Illumina sequencers. I did over 10 billion sequence reads and trimmed the reads and then looked at the 2 different aligners to say, OK, well, what's there? And one of my favorite things about the study, probably the favorite thing, is on average, across all the samples, about 48% of the DNA match no known organism. We don't know who they are. We've never seen them before, except they live on the New York City subway. And so they're hanging around us. We don't think that they're alien DNA. I get this question a lot. Do you think it's aliens? I've seen a lot of weird people in the New York City subway. I don't believe that's the case. It's just actually, there's probably 1 trillion extant species on Earth, and we have good reference DNA genomes for 35,000, right? So we don't have a good catalog of life yet on Earth. And so some of this is not too surprising. We built 656 supplemental HTML5 files, so you could zoom in and out of the maps and see where your favorite species' density is across the city. We worked with the Wall Street Journal to make a map, so if you've ever wondered where all the kimchi and sauerkraut bacteria are across New York City, now you can find out. It's like or other related bacteria, what they're associated with. We also found some really good actors, like Pseudomonas petita is really good at absorbing toxins and chemicals from the environment. It was very ubiquitous across the subway system. We looked to see, well, if you go back to the Human Microbiome Project, what does it look like? Does the subway, we have Staph epi, Staph aureus, Actinobacter, Propionibacter, which is now Cutibacter, acne, things that you would normally find in human skin. If you look at the log ratio, it looked the most like skin, but we also see evidence of the urogenital tract bacteria. I think it's because some people don't wash their hands, so please wash your hands today after you deposit your sad moment of lost stool. Please wash your hands. So this looks most like skin. This was replicated by Curtis Huttenhauer's lab in the Boston subway a year later. They also say it looks mostly like skin. And over 600 species ride the subway with us, and most of them are harmless. Some of them, will kill you. This is Iracoccus viridans will kill you if you're a lobster. So if you're not a lobster, you should be fine. But most of them actually were fairly innocuous, and any pathogenicity markers were usually absent or at very, very low levels for anything that looked like it could be pathogenic. So it was mostly good news. But we did find some other interesting things that were there. I like to call them molecular echoes. Like, for example, in the Gowanus Canal, this is part of Brooklyn, we saw a lot of methanogens that were down here. And it's because there's actually raw sewage that gets dumped into this canal, you can see here. And so it looks like an awful place. Like, you should really never go swimming in there. But one guy did try. He concluded that's not safe to swim in there, is what he said. He put on a hazmat suit, went through there. It's not a good idea. So we found the sewage bacteria. OK, check, great. But then we want to look at other areas of the city. Was there anything unique? So here's some enterics, again, some staph genera. But then Pseudoaltemonas and other species really popped out here. And so that's interesting. It's clearly a different part of New York City. What happened there? And this is actually the station that had been flooded by Hurricane Sandy in 2012. It had 10 species there that we didn't see anywhere else in the subway, or almost anywhere since. And it turns out, if you remember Hurricane Sandy in 2012, came through New York, kind of knocked off the NIH's websites, came through as a big storm, like huge waves coming through. At one point, the surges got so bad in the city, we saw huge waves coming through. I'm just kidding. That's from the day after tomorrow. It's not a real picture. But it felt like that. I had tape on my windows, and actually this is a real picture, though, of the subway that we lost parts of some lower Manhattan, parts of Queens, parts of the Bronx, where the subways got knocked out because some of the subway stations were literally submerged in water. And so the one train did not go uptown that day, right? So it was really completely submerged. So we worked with the MTA to swab the station and see, does it still look like the ocean, or what's there now? And so actually we found species that exactly look like that, including one that was previously found as Antarctica, and it was basically, as you can tell by the name, it's often found in cold waters. And it's also interesting, it actually normally makes something called eicosapentaenoic acid, or EPA, which is normally something you find in fish diets. And so it's interesting, we could see the sort of echo of essentially the ocean water on the walls of the subway station, even a year later, or a year and a half later as well. And if you have low levels of EPA in your diet, actually you'll be at a higher risk for suicide, according to this paper. So this could be a really positive story, where if you do lick a subway pool and this is there, it's actually a good thing. It's definitely not a bad thing, and maybe potentially helpful. So I once had a reporter say, okay, well, Dr. Mason, So is it okay to lick a subway pole? Like, yes or no, I just want to know. And I said, well, it depends on what, I'm a scientist, right? So it depends on what's there. The licking is different from touching. There's a mucous membrane. It depends on what's there, how long it's been there, the temperature, the humidity. You're probably fine, but statistically, but it really depends a lot on a lot of variables. And so the only thing that appeared online that next day was basically this headline. It's basically, which is technically true. Statistically, it's unlikely to be harmful. But I stand by this not only because it's true, but and also as a lesson, they will quote you with only 2 words that I did technically at some point in that long answer say probably fine. But also, there is a great paper that came out the year after our study was looking at, if you think of like what you need to have for exposure in the environment. For example, if you start to become allergic to peanuts, the worst thing to do is to take away all the peanuts from your environment. It's better to actually have low level exposure to antigens, to things that are even that you're beginning to get allergic to. It's actually what you want to do is continue to have exposure for your immune system to get training. And you sort of build your own immune systems network. And also from this paper, this was replicated a year later. You need to basically build up that, basically exposure to the environment, what's called the hygiene hypothesis, to build up your own immune system sort of force field as well as your own microbiome. So that's why I stand by it so far. So that was on one level, on the microbial side. But then also is the human side. So as I've alluded to some forensics features, could we, for example, we were inspired by this study where they recreated the map of Europe just from genetic data alone. You can see the colors matching from John Novembre. And you can see here they claim they could predict within a few hundred kilometers of where you're likely born based on these ancestry informative markers. So all of us here in this room have different eye color, hair color, skin color, based on essentially how humans migrated across Earth for the past 100,000 years. And that's represented in these ancestry informative markers that you could even, in theory, pick out from this map. Or in this case, Heather Dewey-Hogberg, she said, well, what if I took DNA from a cigarette butt and I could just pull out the DNA and pick out the ancestry informative markers and sort of predict what the face would look like to shame the person who had left that cigarette butt behind and make these kind of spooky masks like this of people who had left their cigarette butt. So inspired by this work, we thought, okay, well, let's look at New York City. Here's the map of New York City. You can see here's the census data is Black, White, Asian, Hispanic. That's all they ask, those 4 categories. And you can see there's different parts of New York City. You can see Harlem, you can see Chinatown. This is basically based on census data. It's all open source. You can actually go in and make these maps. And so we want to see, well, there's human DNA left behind. Does it match the census data? So we did a segmentation algorithm to break the city into different segments and then looked at where we collected samples. In this case, sort of a more, on the census data, a white area in Brooklyn. And then we did just a prediction based on the subway bench. What does it look like as present? In this case, it looked much more like a Tuscan, British, Finnish, and European ancestry on the subway station. And then we looked further up down to Manhattan. So this place kind of looks like it's in northern Harlem, more of a Black and Hispanic area. And indeed, it looked like from the DNA on the seat, more African American and Yoruban. Basically, it looked as if it was an African American seat bench based on the people that lived in that area. So it actually means there's a bit of a human molecular echo. And you can see this is the whole data set. At the very top was the deviation was near 0. So we could almost perfectly recapitulate the census data based on the human DNA left behind. In other cases down below, it would get more noisy. But you can see this interesting idea of a molecular echo. And so based on everything I've told you, if we had DNA sequencers set up everywhere, on the toilets, on the seats, on the subways, everywhere, could we actually, are you starting to get a little bit worried about privacy? Maybe you are, because I've been alluding to the fact I'm tracking you everywhere you go throughout this whole talk. And so if you're concerned about this, so there are some legitimate concerns, and GINA is one law that was passed to prevent genetic discrimination from occurring, but it's only for human DNA, not microbial DNA. And so all of us do have a gut print. You have a unique gut signature, which you can track as well. So you might start to get a little bit worried and say, well, okay, if this is true, what can I do? Chris, tell me, what can I do? And I said, well, I have a website for you. You can go to bioanonymous.me. You really have 2 options if you're worried about this. You can either spray DNA everywhere you go when you're leaving the room, or you could grab DNases and degrade the DNA everywhere behind you so you just erase your tracks. And you can actually buy both of these products on this website if you're interested, so you can, if you're concerned about this. I have no financial relationship with them. I guess I just think it's interesting. And, but that's not the biggest concern I get. Most people just say, okay, well, Chris, just tell me, just should I ride the subway? Just tell me, yes or no. And my answer is very much yes. You should do it probably ideally with ice cream, because it's mostly fine. It's probably fine. And then also, it is not an endless cesspool of disease and filth. It does get cleaned. Not every station every night, but most stations each night. So there's the staff at the MTA cleaning. They take Tide and water and basically scrub down the walls, the floors, the benches, the garbage cans, even some parts of the ceiling. So it, essentially does get scrubbed. But if you think about what I've just told you, there's a genetic map of what's present. And what happens is when they go down and clean, it's basically, they're going in, scrubbing away what would have been deposited there for the day. This is basically, the equivalent of a forest fire onto the microbiome of those surfaces. And then every morning what happens is people come back down, whatever they didn't shower away, they'll repopulate it and redeposit their microbiome. This happens as continual diurnal cycle of microbial exchange. And it's not just happening in New York City. Of course, there's other cities that have subways. So this is, for example, one in Washington, D.C. There's a guy who's mopping kind of on an escalator. What's interesting is here, I don't actually even know if this guy works for the transit system. It's just a guy I caught on film, just mopping away. So clearly there is a large context. And we wanted to build was a global context, which some of you are contributing to with the study today. So can we actually build a genetic map not just in one city, but many cities? And also, what haunts me, what pleasantly haunts me actually, is this blue number of the amount of things that are just unknown. It's really who is there, what's there, and what are they making? Can we actually use this information to find new molecules or discover new life? And the answer is very much yes. And so it's also not the first time that we've had half the world unknown around us. When Magellan and Columbus set sail, this is what they thought the world was. Of course, they were missing half of it. And when the microscope was invented, it let us visualize a world that we couldn't see before in 1673. But actually now sequencers are basically molecular microscopes that let us see the world that was previously invisible. So with help from the Gates Foundation, we launched a global consortium and also help with collaboration with Illumina, is to build out these maps with three big goals, is to just map essentially the metagenomic maps around the world, use this as a means to track antimicrobial resistance as well, and look at the trends, and then also to use this to mine the data, look for new antibiotics and new molecules. So what I'd love to do, if I could, is have a giant swab and basically swab all of planet Earth in one big swoop and then sequence it. But I don't have a swab that's that big, and I'm not that tall, so I can't do that. So in lieu of that, what we do as a global consortium, once a year we do a global sampling day, and also we have other some more seasonal sampling that gets done. Basically, as people wake up past the international date line, you can see they'll start to populate on the map, and you can see as the Earth rotates, we actually start to do the collections in synchronous, same protocol, same methods, and now also on Illumina sequencing. So this is sort of the global consortium across all the transit systems to track what's happening in urban and really dense human environments around the world. So a project called Metagenomics and Meta-Design of Subways and Urban Biomes. And you can go on the website, you can look at all the samples that have been collected. You can see the interactive map here. In the past 2 years, we've now gotten to almost 15,000 samples over the past couple of years. And so far, across all the collaborating cities, what have we seen so far? Some really interesting preliminary data has shown that even in New York City subway systems, actually, for example, on metal stairway railings or plastic signs, these are predicted new biosynthetic gene clusters that are likely making new molecules that we can find for the first time. And just as, again, you look at a subway pole or look at like a bus stop and you might think like, I don't know if I want to sit there. You should be excited and wrapped with energy and splendor and wanderlust about the capacity and the amount of biological discovery that you can do just on that seat bench. Now, I think I'm alone in this view generally, but empirically, that's actually what's there is a large amount of unknown things. So in principle, from work that we've done and others, actually, these metagenome assembled genomes have actually given about a threefold increase in the sort of tree of life, basically just discovering entirely new phyla that we couldn't otherwise see because you can't culture them. They exist in the environment. The other thing that we've done is to see, well, can we take randomized samples and predict where you're from? In this case, you can see like Auckland and Hamilton and then randomized samples. There is also a city-specific signature that seems to be present in these data, which is part of what our hypothesis is today, is can we tell if you've traveled from another city, is that still present on your phone? And so that's one of the things we're looking at today. And the other thing, of course, is one of the goals is to build antimicrobial resistance maps. Here's an example map so far that we've generated. This is for London. And you can see the density and picking points of different antimicrobial resistance markers, say tetracycline or other markers. You can see the density of them in log scale across London. And the last thing we're doing, it's a consortium, is that we did in 2016, we swabbed Rio before, during, and after the Olympics. And we're doing the same thing in Tokyo in 2020. So this will build us basically a global map over 5 years, and then we'll see how they move over the course of this entire 5-year study. We're about halfway through. And so what you'll do, if you can, contribute today, if you go outside. So after the lecture, we have a great number of volunteers and collaborators, some from my lab and a lot from BioForward, that'll swab your phone to contribute to what's the baseline information of AMR markers, and also where did you travel from, and is there a signature that's still there on your phone, and also just general biological discovery. So you'll get a little report, and please join the study. It's free, and I promise not to send you creepy emails, even though I've promised in some ways that I could. So I want to close in the last couple sections on some work we've been doing with NASA. So that's on Earth. We'll get eventually an entire genetic map of most of the cities across, of all the major cities across Earth. But what about beyond Earth? We shouldn't limit ourselves to just one planet. So could we think about going farther, as I alluded to in the first slide? And can we think about sending people far away? So this is something that NASA has been thinking about and working on for decades. But only recently did they have a chance to say, okay, if we're really going to study what happens to the body in space, it'd be great if we could do it for a long time, like a year. So NASA launched their longest ever mission, a year-long mission. And we'd want to have a good control. In this case, we were lucky that Scott Kelly had an identical twin, Mark Kelly, that was on Earth, and has identical twin, so it could be monitor them essentially for a year in space and have a ground-based control. And so NASA put out a solicitation, and our lab was one of the 10 selected. And so they said, okay, we're going to need to make you a patch. I said, okay, great. So we've got a patch, and you can see here the twins floating in space. And you can see here, I'm very excited that actually I asked the artist to put epigenetic marks right here on the DNA. You can see little circles. And then I said, could you have RNA coming off the DNA and making it more like active? And the artist was like, well, I think I might make it too busy. And I thought, well, fine. I disagree, but fine. And this is the final patch that was for the study, for the mission. And so Mark launched into space in March of 2018, and so made his way up to the space station, was up there for a year. And I'll tell you a little bit in sort of the closing section about how we did the study. So he was up there for a year, and then when he came back, though, this is the video of him. So this is the Soyuz undocking from the space station. If you can read Russian, it says adios, and we're going to get vodka, I think is what it says right there. It just says it's just telemetry data. It's not that much. But there's the Earth down below, and you can see Scott was making his way back. I was very anxious at home. My wife was like, are you more worried about the samples or the astronaut? I said, both. I'm very anxious, but Scott, he made his way back. This is in Kazakhstan, and had been away from Earth for a year, and then finally got back. I was really glad to see him give a thumbs up when he got back. But when he got back, the thing that he'd said is that first day back was really awful. Basically, all of his skin felt like it was on fire because he hadn't felt sort of the weight of clothing for over a year. So just that weight alone burned his skin and felt like it was on fire. He had rashes and discoloration anywhere that had touched anything. Basically had to walk around the house naked because he felt like he had to go to the emergency room. Some people walk around the house naked. You might call that just like Sunday or something, right? But he did it because his whole body was burning. And the big question we have is, what happened to him? Why was this occurring? What happened inside of his body? So to figure this out, we wanted to study the sort of molecular biology as many ways as possible along the lines of what I just told you. So in particular, if we're going to look at two people and try and figure out what's happening in the body, we want to do it at every possible omics layer. So we want to do genetic variation, epigenetic changes, RNA expression, RNA methylation, proteomic changes, chromatin, antibody titers, B-cell, T-cell counts, telomere length, cytokines, metabolomics, microbiome, vasculature, and cognition changes. And do this, not just once, but actually do this in an integrated fashion, 6 months pre-flight. We did it 12 months during the mission, and 6 months when he came back to Earth, including a total of 19 time points collected, as well as 3 vaccinations. And we did this in 2 ways. Was actually some frozen samples. So there's a centrifuge on the space station. Spin it down. We had the same matching terrestrial protocol. Actually, I'll never forget the first phone call we had with NASA to plan the mission. They said, well, what's your terrestrial protocol for this? And I said, what do you mean it's the terrestrial protocol? It's just the protocol. They were very sort of demeaning me, like, your terrestrial protocol. What is your terrestrial protocol? And I said, it's the only RNA sequencing protocol. There's no other protocol, but it's the terrestrial protocol. But we matched it so that we could have the same centrifuge happening terrestrially and also on the space station. And we also had some fresh samples here. So basically, for doing genomics in space, you really have 2 options. We brought samples back, and we had begun the work in the past year to also do sequencing in space. So in particular, this is the space station, so if you want to collect samples, what it looks like on the inside. This is the toilet. This is where we get our microbiome samples. So when you go to your toilet today, you basically should be very excited that it takes you just a minute to sit down and do your business. Here, it's a 20-minute process. They have to set up the vacuum pump to make sure things don't float away. It's a very complicated process. So go to the bathroom with a feeling of liberty and ease today, please, because the number of hour-long conversations I've had about this toilet are incredible. So, but it's an incredible toilet as far as toilets go. Okay, so I'm going to stop talking about that. So, but there is a freezer on the space station, so you can actually centrifuge them down, plop them in the freezer. It's like a -80. There's also some of the frozen samples we got that are urine as well as blood samples. But we also got some fresh samples. So in this case, we had a blood draw that was done using the Wakata protocol, which is basically how do you draw blood in 0 gravity. You can see here the challenge is the tube doesn't really stay in place. And often they're on an empty stomach in the morning drawing blood on their own, and so they have to do their own phlebotomy. And so basically, it couldn't be less convenient when your tube's floating around, but they actually got it done. Wakata did it, Scott Kelly did it. Sometimes they have help, but not always. And you can see here we'll do basically a CPT or an EDTA tube, and we don't just get one tube, but if you look up there, you'll see other tubes waiting. We'll often get sometimes 7, 8, or even 10 different blood tubes, EDTA or CPT tubes. So it's a lot of blood drawn for all the studies that are done. But so we got some fresh blood drawn, plopped into the Soyuz, and had to come back to Earth. And this is a video of some of our samples returning back to Earth with the retro thrusters firing and samples returning. Got picked up by helicopter in Kazakhstan, then repatriated and brought back to Houston. It's a verb I've never used till this study, repatriation of samples. Came back, and then we purified cells, actually had live cells for ChIP-seq and telomere assays that after only 36 hours actually had over 98% cell viability, 99% in some collections, so it was really pretty good. We could actually see here all the PBMCs, or the mononuclear cells. So what happened? What do we normally lose with age is actually telomeres. Some of the results I'll tell you about now is that telomeres usually get shorter over time. But surprisingly, and actually shockingly, what we saw for Scott was the opposite, actually. His telomere length got longer, you can see here, over time compared to control. So here's, Mark Kelly's right next to him, Scott Kelly, his telomeres got longer. As soon as it got back down to Earth, though, they went away. So gravity sucks is really the answer there of what happened. And we said, was this just him, or is it other astronauts? With 11 other astronauts, we see the same thing. So there does seem to be some interesting aspect of spaceflight where your telomeres actually get a little bit longer. Again, a completely unexpected result of the study, which we've replicated, and so this is in review and it'll probably come out in January. But it is really a really shocking sort of part of the study, which is confounded by these variables of radiation, different diets. So it's a different environment, of course, but this is one of the surprising features. We did also look at epigenetic age, and so I alluded to that earlier, and we could see here that his epigenetic age for Scott in blue started lower, went up a little higher in the mission, and then came back down. But overall, he started about where he left off after 2 years, and so he stayed more stable epigenetically. You can see his chronological age there in red. So they're both still, of course, about 50 years old, but it's interesting, Scott is actually, he had more space flight, and he's actually a little bit younger. So it's interesting, it's only one sample, but it's an interesting trend so far epigenetically. So this led to some interesting headlines where people would say, wait a minute, didn't he also gain a couple of inches because there was less compression on his spinal column, so he actually got taller, he got younger, and sort of telomere-wise and maybe genetically, and then also, he's basically, taller, younger, and he lost weight. So I had one reporter and say, well, Dr. Mason, sign me up. I get taller, younger, and I lose weight. I want to go to space right now. And how much time can I lose? Is it like Interstellar, where you could lose decades of your time in that movie? And so I did the math once, Einstein's time dilation equation on the way to Houston, and the answer is pretty much no. I only get about 0.1 seconds difference, and he was born 6 months earlier than his brother. So really, it's not that big of a difference between the age. So you don't really gain that much time. But the other thing that's interesting is there's other negative parts of being in space and being irradiated. In particular, the number of inversions or low-level breaks in chromosomes and rearrangements keep increasing at a linear scale even when he got back from the mission. So we think there is some evidence of irradiation and damage to his genes. And also about 7% of genes that had changed in flight here, you can see all the genes on this heat map, some of them did not return back to normal after being on Earth for 6 months. So there are some evidence of genes that just are perturbed by spaceflight that haven't returned to normal. And he talks about some of the hypoxia genes even in his book. If you want to read a first-hand account of the mission, you can read it from his book. And then also the microbiome change. So jump back to the microbiome, you can see all the different, each color is a species. So there was a same number of diversity, but the ratio of firmicutes to bacteroides, which is an indicator of gut health, got worse over time. The higher ratio is bad here, and that went away though when it came back to Earth. So there are some stressors for sure in the body that can and do occur. And finally, we do have, I can't release which species yet, but we do have some evidence that when he went up to the space station, we compared our data to what was being done for swabbing the space station. So we know that some species in him got planted onto the space station, and the vice versa has happened. So there is an astronaut's guide to microbe hitchhikers if you're curious to see, and more of that will be coming out next year. So all of us are exchanging microbes all the time. So when you shake people's hands today, be like, oh, nice to meet you. You should kind of be like, nice microbiome, and you can smile at people because you're all exchanging microbiomes at this whole conference. You could turn it into a little fun game. And then maybe put your hand on someone else's phone, swab their phone, email each other later. There's a lot you could do today. It's very exciting. So that's the sequencing here. The other thing we could do is, could we do sequencing in space? So could we just, sequence in space would be easier. And so we started this mission with NASA on our way to doing the planning. As we were sending up some of the supplies, the CRS-7 blew up from SpaceX. It was back in 2015, and it was actually kind of a sad moment. No one died, but we lost all of our supplies. And actually, after this happened, I got a really nice letter from NASA saying, well, Dr. Mason, space is very hard. Basically, I'm sorry we blew up your supplies, keep up your enthusiasm, meet adversity, and keep plowing ahead. And I think that was great because if you get a budget cut from the NIH, you don't get a letter, maybe you kind of get a short email. This was like a touching letter to keep going, and they just blew up our supplies, right? So it was really, I was very touched, and it was great. So we're talking about this on one of the twins calls, and Andy Feinberg, as investigator, said, hey, I want to try these new positive displacement pipettes that should work in zero gravity. Do you want to try an experiment? I said, yeah, we should try the sequencer and see if that'll work. He said, okay, we'll send it along. I'm going to try these pipettes first. He says, all right, I'm going to try this. The parabolic flight simulator lets you do it in 0G for about 15 seconds at a time on a parabolic flight. But you can see the problem here with the pipette tips is that they don't stay in place. So any of you who have done pipetting know this is not what you want to happen for your pipette tips. So he's trying to work, maintaining his exposure, maintaining his composure. People behind him are puking right now. You can see in the video, but it's the parabolic flight simulator. And eventually you get to the bottom of the parabola and things crash back down. But Andy was a great sport about it. This is a great little experiment. I did manage to load the flow cell. We could show that zero gravity sequencing works. We got some data. And so actually then the next astronaut to fly up was astronaut Kate Rubins, who's a trained virologist. And so she safely made her way up to the space station. We set up resupplies up on the next rocket and actually made it up there. And she did manage to sequence in space for the first time 2 years ago. It was called a game changer by NASA. Some people online called it the dawn of genomics in space. This was huge. It was retweeted 4 times. You can see this. That was pretty amazing. I was one of those retweets, you can see right there. Yeah. But it was definitely big for us. And eventually, when NASA makes an announcement, more people retweet it. That's much more important when they do it. So we did sequence 1 billion bases in space, and then actually published this paper in December just this past year. So you could actually sequence and assemble genomes on the space station. And now, actually, new infections will be mapped and characterized in real time on the space station. So sequencing is now almost interplanetary, but not quite. And so I want to close in the last section just thinking about more on a philosophical note about sort of thinking about biology in the long term and even engineering biology. So I think the way we understand biology, or the way I think of it, is that can you tweak something and engineer it and then predict what happens? This is, I think, the ultimate test of our knowledge. And so they call this a lot, thinking about precision medicine in this crowd and elsewhere and in New York to understand health and disease. But I think if you can become more predictive about your medicine, you can then tell whether or not it'll work and then how long it'll work and go back and forth and have it be a virtuous cycle for health and disease. We just launched a new initiative at Cornell called on predictive medicine and predictive analytics. And this is a new institute here. And you can see here, we're just looking at a screensaver right there, but we're looking very intently at the screensaver. But this idea of precisely editing and measuring, used to be almost science fiction until 2015 when genetically engineered embryos happened, right? So this has already occurred. Or just last fall, the idea of repairing an embryo that had a defect in a heart disease, hypertrophic cardiomyopathy, repair the embryo before you actually would implant it. And this guy, he looked kind of smug in this picture, I think. I kind of like this picture. But you can actually do it now. This is, again, science fiction is not really science fact. And also this year, there's $150 million for somatic CRISPR trials, including one that is from the NIH, actually doing somatic tissue editing, essentially to treat beta thalassemia. So can you edit hemoglobin in the adult's body in the somatic cells and actually change them? It's already started. And it's also, Japan just last week said they can allow gene editing tools for research into human embryos. And so this together is, really got a lot of discussion, some controversy. It's also led, though, to a research group called the Genome Project Right. You can see here sort of the first paper on it. And there's a consortium now that's thinking about, I'm one of the co-directors for the steering committee, we're just thinking about how do we do this carefully, how do we do this accurately, and how do we deploy it to sort of, new medical contexts. So one of my favorite ones is actually not just thinking about editing the genome, but editing the epigenome. This is a great, beautiful paper from Rudolf Jana showing that you can repair Fragile X syndrome by changing the epigenome the same way you think about editing DNA. You can modify the epigenome as well. So the canvas of the epigenome is also a place that you can tweak and color, and you can order a lot of these online on Edgene. They're open for anyone to try. And then, and also another place that a lot of editing is happening in T cells. So many of you probably heard of chimeric antigen receptor trials, but there's actually, most of them so far are being done in China. In fact, China is sprinting ahead in the most chimeric antigen receptor clinical trials to date. This is from clinicaltrials.gov. And so we know that there's a big race right now to edit T cells, edit immune cells, to use them for immunotherapy, and really edit any cell in the body because now it's really easy to do. So of course, this leads to even bigger questions. Well, should we modify embryos, or can you have mitochondrial 3-parent babies? So this is something that you can have. Actually, the UK just passed a law that made it possible so you could actually have a 3-person embryo. And again, it was upsetting and unsettling to a lot of people, but actually Doudna is on the short track for the Nobel Prize for CRISPR. I liked her quote about this. She said, people get comfortable with technologies over time. IVF used to be viewed as something abhorrent and mysterious and almost too controversial, but now is standard and accounts for several percentage of all births in the US, right? So she suspects this will be the same. I think so too. But I think there's also a sense of not just the fact that there'll be a normalization for it and also safety around it, but I think there's also a sense of duty about how you should engineer things. So in particular, like in the space station, should we just let things grow as they are and like become a risk for the crew and just let fungus grow on the walls, or should we engineer the microbiome of the space station and other spacecraft so that it can be more protective? I think you should as a function of safety. But then also for astronauts, should we send them away to faraway worlds to be irradiated so that we just say good luck? Or should we engineer them so they have pharmacological protection, physical protection, and then also genetic protection internally, like extra DNA repair enzymes or increased activity of them? This is something that we could do, but should we do this? So I've written and talked a lot about this as an idea, but philosophically, we may not have any choice, because if you send someone there, and really, if they're not protected on every possible level, you're putting them at greater risk, and so you may have to genetically protect them as well. And the only good thing about sending heavily genetically modified astronauts to another planet is that there's already a built-in quarantine protocol. They're on another planet, so if something goes really wrong, at least you've got that going on. But more seriously, it's more of a philosophical idea right now, because to have the hubris to say we're going to edit the human genome or edit entire ecosystems or edit entire planets is something that has to be done very slowly, very carefully, and in baby steps, or even baby steps also with a lot of quarantine first. But it's already, but to think of doing it in Mars, which might seem like it's too far away, is actually would be the second time we're doing it. We already on a planetary scale monitor and characterize and try to attenuate the risks of climate change on Earth, and so this is just now us doing it on a second planet. It's 54 million miles away as of this morning, so it's farther away, but the concept is the same. So I think that's the end of my sort of talk, and I want to thank everyone in the lab who makes this all possible. It's really inspiring, and Heba is here from the lab helping outside. I want to thank also Mark and Scott Kelly, who gave blood, sweat, and tears. You can see this is Scott Kelly. This is their official poster from the NASA mission. You can see they snuck in the Death Star back here, right back there. I want to thank, obviously, a lot of funding. Also, Promega has helped, Illumina, obviously, for funding this event today, and also a lot of collaborators, some in this room. And also, thanks to funding from the Gates Foundation, NASA, World Quant, and NIH. And thanks also to BioForward and Illumina again, and thanks to the lab, and happy to take any questions.
Some questions. Any questions? We have microphones for people.
How many want to go to Mars? Anybody? Raise their hands. Anybody? Nobody? If you ask an elementary school class how many want to go to Mars, every single kid puts up their hand, by the way. It's a contrast.
Okay, I'll start. So Scott Kelly's up there, and you said he did have some damage to some of his, that stayed in place when he came back. So now we're going to go to Mars, and we're going to be damaged. Does your body start to adapt, or do we see ourselves changing then to be able to live on Mars, or are we going to just die because we're just going to continue to be further damaged, and that's it? There's no way we're going to survive.
I mean, I hope we'll survive. I also would probably, a lot of the proposals in NASA review are to dig down below the regolith and actually be protected at least a little bit from the radiation. But the lack of atmosphere, or the minimum atmosphere that's there, and essentially lack of magnetosphere means you're really at risk. And so I think both will occur. There'll be people there'll be too much radiation. There'll be people that will die, and that's happened with every space program. It's a very dangerous activity to get in a basically giant bomb and be sent to another planet. So it's risky. Actually, I have a different slide deck that talks about the overall odds of dying as an astronaut. If your job is just astronaut, the odds of dying are about 13%. It's gotten a little bit better in the past 4 years because no one's died, but it's a tough job for staying alive. But then also, people will adapt eventually. It's not, we're all evolving right now, and so that'll continue to happen there.
Awesome. Get some questions. Do we have the microphones back? Oh.
Yeah, I'll steal the first one. Sure. So I read a headline the other day, and it said, checkpoint inhibitor, revolutionizes treatment of cancer. And so I went on to read the article, and it said that this combination of a checkpoint inhibitor and a standard chemotherapeutic had improved life from 12 months to 14 months. And I thought, wow, 2 months. And I'm sure that 2 months means a lot to people, so I don't want to, but it always seems that these technologies overpromise, underdeliver, and it's more of an incremental change. Just interested in your thoughts on CRISPR, because it seems like it's being so widely overhyped right now, and the reality is that it's got to be far more complicated than it really is.
Well, yes, and it's also, I mean, the idea of it that it's just like a word processor and you can CRISPR anything you want, and that, high school students are doing CRISPR. I feel like, college students are doing it. My grandma's doing CRISPR in her kitchen. Like, everyone's doing CRISPR. But there are unexpected things that we've already seen. I have other slides that talk about this. Think about what you're doing with you have to induce double-strand breaks, and then what ends up happening is actually some of the cells that end up surviving from that might be mutant for p53. So you might end up actually enriching for mutated versions of your cells. So you might accidentally create the worst cells that survive from some of your CRISPR therapies. This is something that's been published. And then also it's not perfect. So even for T-cell engineering and modifying some of the T-cells or natural killer cells, they become resistant, right? So there's immune cell exhaustion. You can't just edit the cell and have it do everything you want for the rest of its time. It will become exhausted or resistant or eventually apoptose. And so it's a tool. It's not the tool, I think. And people working in the field and ourselves included, like, we know that it's a tool. But it's just like PCR. Like, you would never think PCR would save the universe. It's just a way to manipulate the universe, I'd say. And so CRISPR will be the same. I mean, it shows a great promise, but it's not going to be the be it end all. I mean, I actually think, for example, genetic editing is one factor, but you'd also have to layer in epigenetic editing at the same time, particularly in leukemia, which we've published, which evolves independently in some kinds in resistance. So you have to be able to do both. Yeah, I'm done here.
Dr. Mason, that was a great talk. Thank you. So in doing your microbiome studies, is there a sort of an ideal microbiome that you can take the best of microbiomes from, like, say, Japan and the US and blah, 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 and be like, oh, this is the best microbiome to have? And then we can just distribute that to everybody.
So a good question. So it is kind of like, I mean, it's like a probiotic for the environment or the built environment. So there is something called sick building syndrome that there's an ongoing work to see. Like the buildings, when we create them, and even from every room has an ecosystem. So we're already accidentally making entire genetic ecosystems. We just haven't been measuring them or trying to engineer them. So I can't yet say if there's an ideal one. It's like, is there an ideal human genome? Like, maybe George Clooney has got the ideal genome, but like I certainly do not, right? So I think there, but even he has mutations, right? So there's no such thing as the perfect human genome. Or maybe, but there are ones that are certainly have less risk of disease, and so I think you could, begin to do that process of gauging which ones are in building environments or space stations that seem to, be the least likely to grow sort of pathogenic fungi, or just seem to be, generally not lead to any sickness or allergies. And so, but we don't know what those are yet. We've just begun to map them for the first time. So I don't know if you need, 10% Doha, 20% Sao Paulo. Probably need like 5% New York in there, just a little bit, just to get like the grittiness in there, I think. So the short answer is we don't know yet because we're making the very first maps. But eventually, I think that would be the goal. Yeah.
Dr. Mason, if we're going to be on Mars in 2035, how far along is NASA or the CIA with genetically editing our astronauts? Or is that already happening in the subways with that unknown species?
So how far? So I can neither confirm nor deny any evidence of any engineered astronauts, but most of it's still in discussion phase. A lot of the technology is very new, and the ideas are also, I think because there's actually, for every NASA proposal, they have something called the technology readiness level. You have to say, well, how close is this? Is it like a 9 where you're ready to send this right to the moon or Mars right away? Or is it something that's early stage? So most of the technology that they've rated so far for some of the CRISPR tools are still early, so like in the 3 or 4. So I think that from their perspective, and they've said this, and it's in some of their documentation, is that they're excited about it, but it's still in the early stage. But I think it'll start first more with microbes. We'll engineer the microbes. They're easier. And maybe eventually larger species, but the astronaut engineering is something probably still 10, 15 years out, I'd say. Except I've got there's one island in the Caribbean, but I can't tell you which one, where we've got it all done, and some Michael Crichton's going to be there, yeah.
Dr. Mason, that was an incredible talk. Over here.
Okay, good, yes.
Miguel with Mirrors Bio. I had a question. You were able to essentially quantitate how many organisms were unknown in the metro system. And my question is, how are you able to identify between unknown organisms, first of all? And then second, how would we use that information to find them in the future?
Right, it's a great question. So how do you know? It's like remember the Rumsfeldian known knowns and unknown knowns and unknown knowns and known unknowns. Unknown unknowns, that was a great one. These ones are just unknown because the DNA we know is of good quality. It's like an Illumina read that is of good quality, like the sequence is of good quality based on the reaction. So we know that this DNA came from something on Earth, maybe from not on Earth, actually, but it should be on Earth. And the sequence is fine, the chemistry is good, so that molecule is right. It's just when you try to map it to a database, it just doesn't match anything. And so the two ways we go around that is to continue to work with other consortia. So there's something called the Vertebrate Genomes Project, which is trying to create a reference genome for every living vertebrae in the next 5 to 6 years. So some of the DNA will be different species of insects or rats or other things that were in the subway that eventually will map those reads to the reference. But right now it's like trying to go to the library and you think there should be 1 trillion books in the library, but there's only 35,000 of a reference that you can look up and even try to match something. So it's really just a database problem is the biggest reason. But the second way we're getting around that is doing what's called de novo assembly. You link together the reads to make longer, what are called contigs. Then those you can actually sometimes align them or align a piece of them. And so you get a partial match. But much of it is to find entirely new things. So it just means if it doesn't match any known database, that makes it potentially interesting because it's coming from a species that's either unculturable or just not been sequenced yet. But it doesn't mean it hasn't been seen yet. It could be, something that just is a normal flora or fauna of that area that you see every day, but just don't have a reference genome for it. So, and you all have some on your phones as well. So if you do the phone experiment, you all get a little bit of how much mystery you have, and we could have a prize for who has the most mystery, which maybe would be the bathtub full of yogurt. I think is what you'd get. I think. All right. Great. I think time. Thank you very much.
Big round of applause.